Because obviously there's not time to do every single one. So this way, if you're confused about it, do you think you're the only one that couldn't figure it out? Probably not. But I don't know which ones you don't know. Who knows? I think you know all of them. Maybe I'm not right. I don't know. We'll find out. So there wasn't enough stuff on that study guide to make a quiz for today. Don't be too disappointed. So instead, we'll do something silly to start with. Oh, right, we'll do that one too. Let's do the silly thing first. So what we're going to do, this is one of those little illusion things. We're going to look at that image for 30 seconds, focusing on the 4 little dots in the middle. I'll point to them. And when I tell you to, then you're going to look at some white wall, some direction. So not out the window, but like up here, over there. And then wait till you see a white circle of light and then blink a bunch of times and see what you see. All right, so 30 seconds. Do you see those little 4 black dots in the middle? So just stare at those. Don't think of anything else. It's like a meditation class. Nothing's going to happen while you're looking at it. It's not one of those scary things where it jumps out and gets you or something. That would be mean. All right, so then look at a white wall until you see kind of a circle appearing, and then blink a couple times. Do you see anything? Yeah. It's not weird. Don't ask me why that happens. So do you see how many people saw a face in the wall? That's not weird. There you go. There you go. It's funny, that's what everybody always says. It's interesting. So if anyone has, after we do this chapter, if you think you figured out how this works, let me know, because I can't really figure it out. I almost have it in my head, but I can't quite put it together. If you really want to make yourself feel crazy, just type in optical illusions on Google Images and just click on one after the other. Your brain's like melting down. All right, so we have one question left to do from last time. That was number 4. So I'll let you do that with your group. Does anyone need one? I've got some extras. All right. So what did everybody come up with? So a big part of it is the olfactory system, because what we think is taste is mostly smell. What's the second answer? Yeah, so like different combinations and like different amounts of stimulation. So like, what are 2 things that are sweet, for example? What's something that's really sweet when you eat it? So like honey. What's something that's just sort of sweet? Okay, so like strawberries or something. And you can tell your taste buds are sending more signals from the honey, the sweet ones, than they are when you eat the strawberry. And then maybe there's some, combinations of the sour as well, in the strawberry, depending on how fresh it is, right? So it's like different combinations and then different amounts too is important for that. All right, we'll put away the creepy face. I put a few more illusions in later, just for fun. Let me turn one of these on. All right, so hopefully this won't be too complicated, especially because you dissected an eyeball yesterday, Monday. So now we'll go over it. So the sclera is on the outside, and if you look at somebody's eyeball, what color is the sclera? Well, it should be white. Maybe it's a little red if, allergies or something else. Behind the sclera is the cornea. The cornea starts to focus the light a little bit. And then the pupil is the hole. And then the iris is the muscle around it. So the colored part of your eye is the iris. And then it goes through the lens. You saw the lens in the cow eye yesterday. It was pretty big and hard. Because it is a lens, so it's hard, like a lens, like glass. And then all back here is your retina. That's what receives the light info and kind of converts it to electrical signals and everything. And then, so all those neurons eventually form up to make the optic nerve. So you have a whole bunch of axons going through there.
So when you hear someone having a detached retina, is it that it's like actually peeling off the wall of the inside of their eye?
Yeah.
How does that happen?
I don't know. I would imagine trauma, but probably just randomness too, some random weird problem. Because I mean, it's not like if you scratch your eye or something, like that's way out here. You're not like damaging your retina. Yeah, you do hear about that. And we will get into some of the other eye problems like farsightedness, nearsightedness, astigmatism. All right, and in case you want to see a slightly more complicated version of the eyeball, it's basically all the same. The one thing this one has is all that adipose tissue that you saw when you dissected it. So you could see, what do you think that adipose tissue is there for? Protection from what? It's like movement. Something hits your eye's kind of moving. It's got all that cushion around it so it doesn't get damaged. And there was a lot, right? There was a lot of fat around the eyeball. That was not because it was a fat cow or something. It's normal. And then you also have all these muscles. What are some things that those muscles do? What's their job? Open and close your eyelids. Move your eyes side to side, up and down, rolling your eyes if you need to. When you're moving your eye side to side, like why do you do that? Yeah, but why do you need to, I guess is the question. What part of the eye are you pointing at what you're looking at? You're pointing like the pupil at it, right? That's kind of the idea. Maybe that's obvious, I don't know. All right. So one thing you may not know is that the default setting for your eye is to focus far away. So over 20 feet away. That's just how it's set up. And if you need to see something closer up, that lens has to adjust its shape so that you could focus on something closer to you. Just like a camera, right? If you're like turning the focus thing, it's like focus far away, not close up. Turn it, and it kind of changes to be focused near but not far.
So then when someone gets older, does that adjust, suggest the lens such as part of it?
Yeah. Actually, going to get to that. But yeah, we can talk about it. So do you remember in that little experiment you tested your near point of accommodation? Like what's how to describe it? The closest you could get where it was still focused. It's best in kids, and then just after that it gets farther and farther. The lens isn't able to adjust as much. I'm not sure we know exactly why that is. But that's why you see like older people looking at their phones like this, pushing buttons. I know. And so this one's cool because you can actually see that lens changing shape. So that'd be looking far away, looking close up. What would you say changed about that lens? How would you describe that? Condenses. Kind of got squished and got fatter. So a thicker lens, does that bend the light more or less? More, right? Which is why when you get thicker glasses, you'd say your vision's worse because the light needs to be refracted more. All right, so in order to see something clearly and not blurry, those rays of light coming from the object have to meet at a point right at the retina. And once again, just like a camera, when you're turning your focus thing, you're actually moving where that focal point is, like where that point is that it's focused. Same thing with a microscope. When you're moving it up and down, you're trying to get that point at the right spot to get it to be not blurry. All right, so if you have myopia, your eyeball is longer, so that focal point is actually in front of the retina. So things are blurry. So that's like this, and this, of course, this is nearsighted, which means what? What does nearsighted mean? Can't see far away, you can't see close. So you're looking at something far away, the light refracts, and the focal point is like in front of the retina. So that when you look out there, everything's like blurry. But if you put a lens there, concave lens, it spreads those light waves apart a little. So now that when they're refracted, you get to a point right at the retina. And you could see it's not blurry anymore. So if you need glasses for looking far away, that's what kind of lens you have. You may be able to actually look at your glasses and tell. Not sure. And then, of course, we have the opposite. So the eyeball is shorter, so that focal point is like behind the retina instead of on the retina. So we use a convex lens to fix that problem. So you could see it focusing like behind the eyeball. And if we focus it a little extra, now it's going to be focused on the back there. So this is farsightedness, which means you can see far away. Close up. All right. So just a little survey here. All right, so let's see, normal, nearsighted, farsighted, and then E is more than one of the above. So an astigmatism is something sort of different from these. It's like some weird irregularity in like the cornea or the lens that just affects like part of it. So it's different on different people, but still kind of same idea. It makes the light refract the wrong way. It's not focused. But comparing near and far, we got a lot more nearsighted. And that's really common until you get to be older usually. Most people need glasses for seeing far away because it's some weird kind of problem they had started off. But their lens is still able to change shape easily when you look close up. Can you be nearsighted and farsighted? It's not the best word for it, but you can have problems looking far and looking close.
I think you're colorblind.
What kind of glasses would you get for that? So the bifocals. So the bifocal has concave and a convex lens. So you could look out this way and get fixed, and then if you look down, you're looking for through the other part of the lens that helps you read. Those are your bifocals. Anyways, we don't need to know too much about that. It's just interesting. But what I want to spend most of today on is actually the how vision works. I think it's kind of interesting. It's good practice on neurons. So one important thing to know, here's your visible light spectrum. You've probably seen this before at some point. What's different about the different colors? Their wavelengths are different. What else? The amount of energy they have is different too. So like a photon that's blue has more energy than a photon that's red. Which is kind of interesting. And that's actually going to explain how your receptors can respond to different colors of light. It's because they respond to different energies. So like the ones that respond to green, they respond to that much energy in a photon. But if a green one gets hit with an orange photon, nothing happens because the energy is not the right amount. All right, which of these has a longer wavelength, red or blue? It looks to me like the red wavelength is longer because it's the distance from one wave to the next. So it's like, what does it say? 1.5 times longer than the blue. So this is a cool little map to show you what you've got in the retina there. Probably read about in the chapter. You got rods and cones. Cones are for color vision. Rods are black and white. But they are scattered all over the retina because obviously you want to be able to see all the colors and see in low light, like get a full picture. You wouldn't want to just put your red cones over here and your green cones over here. They're all spread everywhere. So even if you zoom in, you kind of see cones and rods of different colors, just all spread out everywhere in the retina. So what do you mean exactly? Yeah, it's more what happens after these are stimulated, like in the next layer of cells. It's pretty confusing, but usually the cones are working right. It's just the next neuron in the sequence isn't functioning right. All right. So you got your rods that are for grayscale, so they're more sensitive. So you've probably noticed before if you're in like a really dark room, do you see any colors really? Or you just kind of see like shapes and gray and black, right? That's because the rods can get activated by a lower amount of light than the cones do. So then cones. Red. So you got blue, green, and red. And these are so you need more light to see in color.
So have you heard people say that babies see colors, or at least not as well as adults?
Mm-hmm.
Is that true? Do you know like why that would be?
I mean, I know they always say babies don't have that good of vision, but I've never actually like looked into what that means. Like, is it color? Is it blurry? I always just assumed it was blurry.
That's what they have to do. Is like, remember when they have like 420 vision?
Oh, 420.
It gets less blurry.
But do you know anything about the color part of it? I wonder how you'd be able to tell for a baby, like what colors they could see. But it's interesting. I mean, yeah, to look that up. Because I'm not sure how you would know. You could come up with experiments. Well, I think you could come up with ones where they're like pointing at different colors or something, but not when they're like a month old. Probably can't do much. All right. So this is what we're going to spend most of our time doing today. All right. So you've got a long series of cells that are sort of in this pathway of, eventually sending a signal to the brain. So we start over here. The retina is receiving all the info, and then eventually it's got to get over to the brain to be kind of interpreted and conscious, so you know what you're seeing. And the first cell there is a photoreceptor. So we'll kind of just draw it on here. I think I lost some letters there. So the photoreceptor cell, when it's not stimulated, so like in the dark, it has sodium channels open. So sodium comes in. What does that sound like? Sodium comes into a neuron. What's the word for that? Could be exponential. You could also say depolarize. Means basically the same thing. So that means, is this cell activated? So it's active in the dark, a little strange, right? It's kind of backwards. So it sends that exponential down, and then at the end of that photoreceptor, it opens calcium channels, and it releases an inhibitory neurotransmitter. And the next cell in line is a bipolar cell. So do you think that cell's being activated in the dark, the bipolar cell? Who's catcalling at me? So inhibitory neurotransmitter, what does that mean? Do you remember from chapter 12? Does that activate neurons or inhibit them from being active? It's going to inhibit them, right? So the photoreceptor cell, it's activated to tell the bipolar cell to not be activated. Kind of weird. So when this photoreceptor cell is activated, it's activated to send an inhibitory neurotransmitter to the bipolar cell. So now this cell is not active. And then the last cell, so cut off here, our ganglion cell. Do you think it's going to be activated? So it's not. How do I hide this thing? Why doesn't it go away? Oh. So the bipolar cell, its calcium channel is closed, and it's not sending out excitatory neurotransmitters. Sharpie. All right, so that's in the darkness. And I actually have a copy of these two diagrams to give you for today. Maybe I'll just pass that out now. Roz, cool. I don't think I passed out enough over here. Please. All right. Nice. There you go. All right, so then what happens in the light for the rod? So if you turn the light on, that causes sodium channels to close. So closed sodium channels. What do you think? Is this cell activated then if you close the sodium channel? It's not active. It's like the opposite of what you would think. So causes the sodium channels to close, so no action potential, no calcium channel open, no inhibitory neurotransmitter. So then the next cell in the line, the bipolar cell, do you think it's going to be activated then?
Oh, yeah.
It is. So it's set up to automatically activate if it gets no signal. Because you don't see any signal coming from the photoreceptor cell. You just see nothing. So then that cell activates, depolarizes, opens up calcium channels, releases excitatory neurotransmitters. What do you think that's going to do to the ganglion cell? What do it? Yeah, so it starts an action potential, which would send a nerve signal through there.
Like, is this just about vision in general?
Yes.
Like, multiple problems is color vision. So this color vision works the same way.
Yeah, it does. So what color, let's say this is a green cone drawn up here. What color light would cause this photoreceptor to close the sodium channels if it's a green photoreceptor? It's green. Now if red light hit a green cone, the sodium channels would stay open and it'd be like it was in the dark. So this diagram would also be a green cone exposed to some other light besides green, like blue or yellow. Something like that. All right, so I decided, came up with a little activity like 5 minutes ago to try. So I need 4 volunteers that are good at talking loudly. Just joking. You could be any volunteer you want. Photoreceptor, bipolar. What else? Ganglion. I will randomly call on people. All you're going to have to do is whisper to somebody else. All right, bipolar. All right, 2 more. 2 more. Ganglion. Brain.
I'm doing it, but I'm not wearing
So we probably need to line up from like photoreceptor over here to the brain on the other side, because we're going to be telling each other signals. All right, so if you would, you can hold up your sign there so everybody knows who you are, your different cells. All right, so let's say we're in the dark. What is the photoreceptor cell telling the bipolar cell? So basically, you're going to be sitting here going, stop, 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 over and over again. Are you going to do anything in response to that? Is it bipolar cell, or you just sit there?
You're gonna stop.
You're just going to sit
I'm
there.
just gonna stop.
Then are y'all doing anything over there? Just hanging out, right? All right, so let's try it. You want to do it once? In the dark. So nobody's saying anything else. Now let's turn the light on. So turn the light on. No, you actually don't say anything, right? Photoreceptor's not doing anything.
Okay, because the inhibitory thing's gone.
Yeah.
Now I.
Well, it's just a signal, right? Like active, signal, I don't know. Well, what do you do? So you're going to tell the brain that there's light or something? Go to the light. So anyways, you can think of it as like 4 people in a row. Each of them's a cell, like saying, activate or don't activate or stop. Does that kind of make sense? Okay. A round of applause for our great actors. I think they need, I think we could use some rehearsal before the actual, maybe like telling you what you're going to do beforehand.
We didn't get some instructions.
That was more like improv, I guess. All right, so hopefully that helps a little bit, because I've got quite a table for you to fill in. Look at this thing. 2 sides. All right, so this is going to take you through every little step on that diagram, more or less. But I only want you to do number 1 first to make sure you're getting it all right before you go on to all the rest of them. Because do you want to erase a whole table, the whole page? Probably not. So all the answers you need are right here. So chat with your group if you get stuck. This is supposed to take a little while because there's a lot of steps and active, not active. Just kind of follow it through. All right, so take a look at this to see if you got it right. Obviously, they're all the same because there's no light. So you can just look at the first, the top one. Am I showing all of it or am I missing something?
You're missing the last one.
I don't want to give away any answers for below. All right, there we go. So did you get them?
Yeah, it was right.
Oh no.
And
Yeah, so y'all got it?
yeah,
Yeah,
I didn't get none of these wrong, just these 2.
just those 2.
Yeah.
All right. So now you're going to go on through 2 through 4. And if you want to put a little note for number 2, when I say low light, I mean like a darkened room where you can't see any color. Like when, yeah. So you can't see any color, but you could see shapes and stuff. All right, so here's the answers to number 2. You can check your number 2. It's a lot easier than me trying to read all these off. All right. You ready for number 3? there's number 3. Although the number 3 disappeared from it. That is number 3 there. So that's something red. That's 3. You're looking at 4. Okay. So one thing that some people were getting confused on for this one, as long as there's enough light for you to see color, that means there's plenty of light for the rods to see movement and shapes and everything. So anytime any of these cones are active and closed, sodium channel, so are the rods.
So like their natural state is to be activated? And then
Yes.
basically when you're not using them, then inhibitory neurotransmitters release.
Right.
And that's what makes them not active when you're not seeing that.
It's very, very backwards. So you got to think that just that first cell is always, unless it's, stimulated by light, it's always telling the next one to stop, 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 don't do anything. All right, and here's number 4. All right, so just to go over that one last time, I can summarize it, hopefully. Keep you from totally giving up and screaming at me. Up a little. All right, so let's see, let's make green. All right, so we've got like our series of cells there. So retina, photoreceptor, bipolar, yada, yada. So maybe we'll do the dark first. That's the one we're working on. So the darkness, does that turn on the photoreceptor cell or turn it off? Yeah, so the darkness, does that tell the photoreceptor cell to make an action potential or not? You're skipping ahead a little bit. So it does activate this cell. Activates the photoreceptor cell. Activate photoreceptor. And then the photoreceptor, what is it going to tell the bipolar cell? So it tells the bipolar cell to stop. And that's kind of the end of it. Because if the bipolar cell is being told to stop, does anything else happen after it? It doesn't, right? So no signals getting to the brain. Is that going to confuse you if I write that, or does that make more sense? Okay, so no signal to brain. Now in the light, what are we doing to a photoreceptor cell? So inactivate. So then the photoreceptor what does the photoreceptor tell the bipolar cell? Does it tell the bipolar cell anything? It doesn't tell it anything. You take away the stop signal. That's what you're doing. Yeah, so now our bipolar cell, what's it going to tell the ganglion cell? Like activate, send signal. And then what's the ganglion cell going to tell the brain? Is it going to tell it there's a signal? Oh. And the important thing, one of the important things to remember then, is those cones, they only respond like they're in the light if it's the right color. So if you're a blue cone in red light, let's see, blue cone. So if you're a blue cone and you're looking at something red, is it like you're in the darkness or like you're in the light? Like in the darkness. Because to that cone, that's darkness. Because it only responds to blue. So that's like you're in the dark. So when you're looking at this thing, that's kind of how you could remember it. So this is... if I ask you a hard question, yes. If I ask you an easy question, no. I don't know what question I'm going to ask you yet. So.
To be determined. Okay.
But you're right. If it's like a hard question, like just, yeah. But if it's a question like, what are the 3 cells in the pathway? Do you think I'm going to give you that to look at? If I ask you, describe the whole series of events in blue light, yada, yada, do you think you'll get this? Yeah. But as you know, you still have to understand it, because just looking at it, did that really tell you how it was working? It's pretty confusing, right? So probably not a bad idea to keep one and look at it. All right, I know that seems confusing for now, but once you look at it again a couple times, it will eventually make sense. And it's confusing because it's backwards from how our brain thinks it should work. There's no amount of explaining I can do. It's backwards. Like, that's all I can say. Different from olfaction and gustation. It's like the backwards system. All right, so I'm just gonna, if you want to look at some illusions, they're there. But I think we just have a few things to talk about for the ear. There's not too much on the study guide for that.
So is our test like the final will be chapter 17 and everything else?
Yeah, so your test will be chapter 17 and everything else. And I think we'll do 20 points from chapter 17. So 20 points from chapter 17 and then 80 points from everything else. So I don't know how many chapters we end up doing. know what that comes out to be. I'm not sure how many exactly. 13. Yeah, so was that 7 points for each chapter or something? Something like that. So, anyways, and another note about studying, like if you come to something and you just think it's way too hard and complicated, ask me, ask the review. But if you just feel like it's just way too much, maybe you're better off just saying, I'm not even going to try on that one and work on the other ones that I actually feel like I know. Because how many questions can there be from one of those old chapters, It's like, oh, maybe I missed 3 points, but if it's going to take me 3 hours to figure it out, maybe you want to focus on some of the other ones. Or at least save it for last, Like wait till you know all the easier ones or the ones that make more sense.
Is it gonna be a written plan, or is it online?
No, it's written. So it'll be similar to your old ones, probably a little more multiple choice than short answer than you're used to. Like what were you like, 70%, 30%?
Yeah.
So it'll probably be more like 80-20. Yeah, we can. It's kind of fun. Yeah, we'll have time. Or at least I think we will. If we have time. If it's like taking way too long, then we can't.
So is the so many different shaped colors, does that come from just kind of like the taste and deodorants, just different combinations
Mhm.
of
Right.
amounts of white?
Yeah. All right, so the ear, just going to keep it simple for there. The ear that you see is actually called the auricle. If you're wondering. And what's crazy about it, so sound comes in. Look, there's some earwax right there. Labeled on there.
That's exactly what it was like.
Just like that. Just one little spot, just clear otherwise. So sound comes in, and then what's interesting about the ear is there's this membrane here that vibrates according to whatever sounds are coming in. And of course, different sounds, they're different frequencies, right? Different wavelengths. And then that little wave is transmitted through 3 bones. So like the sound wave is vibrating the bone against each other, and then the bones actually generate fluid waves in the inner ear. So like, have y'all been to a wave pool before? Maybe that's more in this house. So like, they've got that like thing pushing on the water to make the waves. That's like this bone pushing into this fluid in here. So if it's a high frequency, do you think that's going to be moving faster or slower? It's going to be moving faster, so those waves are going to be really close together. And if it's like a deep bass, then it's going to be making slower waves through here. And so within the cochlea, you have like nerves that are sensitive to different frequencies of the waves going past. But don't need any details of that because we're basically out of time. What did I put on the study guide for this one? Just know the structures of the inner and outer ear or something.
The sound waves to get to the cochlea, structures of the breathings.
Yes. So the pathway and then what's in each one. So you got the outer ear, you've got the middle ear. It's got all the stuff in there. So they've got the 3 bones. Do you know you had 3 bones in your ear in the middle? All after each other. And then you've got the inner ear, which is basically the cochlea for the most part, down there in the middle.
And your eardrum's just that membrane, right?
Yeah, so the eardrum is the tympanic membrane is the fancy name for it. That's your eardrum. So has anybody punctured their eardrum before? So I've heard of, I think I had a friend or something in like middle school, and he's like cleaning his ear with a Q-tip, and he like poked it too far. And you just poke right through that.
I did that now. I like squeaked it, but I didn't actually puncture it.
Yeah, it seems like you have to hit it pretty hard to like puncture it. Did it hurt?
Yeah. I was like bleeding my ear.
Seems like everything we bring up, you've got something.
I was cleaning my ear and I tripped over something.
Yeah, so you should only clean yours when you're sitting down.
Or you're not actually supposed to stick those things inside your ears.
Oh, I didn't know.
But I can't do that. I have to stick my ears.
All right.
I'm like OCD about it.
So we'll do the last the path the sound waves take next time. Just take 10 minutes to review it instead of trying to rush it. But now you got light down, maybe, sort of, vision.